Hello, today we'll be discussing the relationship between serum amylin level and epilepsy by Benlier, Oza, and Orhan. Amylin, or islet amyloid polypeptide, is a peptide hormone comprised of 37 amino acid residues. Amylin is derived from pre-pro-IAPP, a protein 87 amino acids long, after it is cleaved at the amino terminus and processed by the enzyme prohormone convertase 2. This occurs in the pancreatic beta islet cells, and as such is co-secreted with insulin. Studies have shown that the high concentrations of amylin cause aggregation, resulting in amyloid plaques seen in Alzheimer's and type 2 diabetes. Consequently, the co-secretion of amylin with insulin results in high concentrations in the serum of individuals who consume diets high in fat and carbohydrates. Aggregation of amylin and formation of amyloid plaques begins with the formation of monomeric amylin into oligomers. Amyloid formation is almost always preceded by an initial phase, where monomers begin to disappear in conjunction with the generation of oligomers, with no mature amyloid fibrils present. The mechanism surrounding the formation of mature fibrils is not quite known yet due to the kinetics pattern. The abrupt initiation of the exponential phase indicates the presence of secondary processes, and the identity of the kinetic nuclei is not quite known. Studies have shown that nucleation may involve conformational changes in the oligomer fraction when they are present in the exponential phase. However, spontaneous generation of mature fibrils in the absence of oligomers may involve pre-equilibrium of the reaction nucleus with a monomer pool. Fu et al. Proposed a model of oligomeric amylin toxicity involving the amylin receptor AMY3. Increased amylin oligomer binding to AMY3 may induce a signal cascade that results in neuron apoptosis, presenting AMY3 as a possible therapeutic target for the treatment of amyloid neurodegenerative diseases. Epilepsy is a neurological disease defined by the recurrence of spontaneous seizures. Seizures are disorders that involve uncontrolled, hypersynchronous neuronal discharges in the brain. Seizures are mostly time-limited and are usually self-limiting with no need for medical intervention. Epilepsy and seizures are non-interchangeable terms, as epilepsy is characterized by spontaneous recurring seizures, while seizures by themselves are often a symptom for various neuropathologies. The onset of epilepsy may be induced by any factor that results in structural or functional changes to cerebral neurons. Examples include cerebral neoplasms, Alzheimer's disease, stroke, meningitis, trauma, and others. Seizures caused by epilepsy can be grouped into subtypes. The first generalization of seizures is determined by loss of consciousness. The loss of consciousness during a seizure is classed as a generalized seizure. Retained consciousness during a seizure is classed as a partial seizure. Partial seizures can be classified further into complex partial or simple partial, depending on alteration or non-alteration of consciousness. The type of epileptic seizure varies in terms of treatment response, so it is important to categorize the symptoms of an individual to determine the best response.
There is a significant proportion of the Australian population that requires annual epilepsy diagnosis. A specialist organisation outlines current epileptic diagnostic criteria as provoked or unprovoked seizures, with a classification of which can lead to the differentiation of the patient's epileptic syndrome. Another crucial factor to note is family history of seizures, as epilepsy has yet to be definitively linked with inheritance. Albeit the most significant tests involve neuroimaging, as they depict alterations in brain tissue composition, arrangement, and activity, and allow for better differentiation of epileptic syndrome. Abnormal EEG readings of minute electrical impulses are indicative of both epilepsy and seizures, where PET scans differentiate the cerebral areas responsible for the seizures. According to a TAN study in 2014, CT scans are routinely executed to rule out potential presence of tumors, cysts, and cerebral bleeding, as these can also cause unprovoked seizures unrelated to epilepsy. Elucidation is required for the amyloid beta peptide before the human amylin neurotoxic pathways can be properly analyzed, as evidence from a Lim et al. Study in 2008 indicated the two peptides to share the same neurological pathway when aggregated in the brain. For this neurodegenerative pathway to be established, Firstly, the peptide cerebral receptors must be identified, as drugs can be utilized to inhibit this interaction, prevent their neurodegenerative effect, and can be effectively used to map out their toxigenic cascade mechanism. In general, the association of amylin levels in neurodegenerative disorders is poorly explored, as most studies are qualitative and focus on the toxigenic potential of human amylin in Alzheimer's disease, where this article's results can be used to establish a new factor in epilepsy diagnostic criteria. The general aim for the Benlier et al. Study is to investigate serum amylin levels between epileptic patients and a healthy control group in order to provide evidence for the relation between elevated amylin levels and newly diagnosed epilepsy.
The study was performed on 105 patients, 45 newly diagnosed epileptic subjects, and 60 healthy control subjects. The subjects included those that had no previous diagnosis of epilepsy, no prior use of anti-epileptic medication, no prior knowledge of drugs used for neuropathic pain treatment, and excluded those with diabetes mellitus, those that had been treated with anti-diabetic drugs, patients with an abnormal brain magnetic resonance imaging, patients regularly taking chronic condition medication, and those with a neurodegenerative disease, such as Alzheimer's disease. These strict guidelines were decided upon the basis of known variables that would affect the amylin levels in patients from previous studies. For example, diabetic patients were excluded due to it being known that they are amylin deficient due to this hormone normally being co-secreted with insulin, which they lacked. The patients were newly diagnosed as epileptic as part of the study by using routine workup including EEG scans and blood biochemistry tests. During this diagnosis, the epileptic subtypes were disregarded for this investigation. For this study, routine blood analysis and EEG scans were performed for all patients, with brain magnetic resonance imaging being conducted on the necessary patients. In order to analyze the serum amylin levels from each patient, they collected 5 cc venous blood, isolated the sera, and stored it at 80 degrees Celsius. The patient's sera levels were then analyzed using an enzyme-linked immunosorbent assay. The results of this study are clearly summarized in these two tables. Using statistical analysis, a Kolmogorov-Smirnov test showed a normal distribution of body mass index and an abnormal distribution of age and plasma amylin levels among the study and control groups. They used a Mann-Whitney U test for variables with a non-normal distribution and a student's t-test for variables with a normal distribution. A chi-square test was used to analyze gender difference between study groups. From Table 1, which displays a comparison of study variables between the epileptic patients and control patients, there was no significant difference between study groups for gender, with a p-value more than 0.001. The BMI is statistically significant, with a p-value less than 0.001, and shows that there is a statistically higher BMI in the control subjects compared to the epileptic patients. The age comparison is not statistically significant, with a p-value more than 0.001. The amylin levels, however, were statistically significant with a p-value less than 0.001. The amylin levels were much higher in the epileptic group, the median being 226.62 nanograms per ml compared to the 103.66 nanograms per ml of the control group. For Table 2, it shows the association between serum amylin levels and gender among epileptic patients, to which there was no statistical significance, as the table shows that both the epileptic group and control group have a p-value larger than 0.05, being 0.5 and 0.2 respectively. From this, the most significant results is that the epilepsy group showed significantly higher amylin levels, whereas the control group showed significantly higher BMI.
The study found that epileptic patients had significantly higher serum amylin levels compared to non-epileptic controls, with a p-value of less than 0.001. These results demonstrate evidence of a link between increased serum amylin levels and epilepsy, potentially providing insight into the pathogenesis of the disease and opening up possibilities for new treatments. The study also found body mass index to be higher in the non-epileptic control group, who had lower amylin levels compared to the epileptic patients. This supports previous literature showing an existing link between obesity and decreased amylin levels due to its crucial role in glucose metabolism and regulating food intake. The experiment was valid as multiple variables that are known to affect amylin levels were controlled. The study only included patients with newly diagnosed epilepsy who had not yet undergone treatment with anti-epileptic agents. Subjects who were excluded from the study included those with existing conditions such as diabetes mellitus, Alzheimer's or Parkinson's disease, along with those who exhibited abnormal MRIs.
Whilst previous studies in this field explored the toxigenic mechanisms of amylin plaques through utilizing much more advanced and tailored enzyme-linked immunosorbent assays and imaging techniques on rat hippocampal and cortical neuron cultures, the overall biochemical methods utilized by Benlier et al. Were found to be standard for their objective. This was concluded from previous studies with similar objectives of investigating the association of amylin concentrations to varying disorders, as they employed the same kit-derived immunosorbent assays and controls, along with similar treatment and storage of their patient sera, and overall employed similar imaging techniques such as EEG and MRIs and statistical analyses, albeit as Zhao et al. Study accounted for additional blood analytes that Benlier et al. Study did not, such as blood type with glucose and lipid profiles. The neurotoxic potential of amylin aggregates is not a new concept. It has been previously explored due to the pathophysiological implications of its similar shape and function to toxic amyloid plaques. Previous studies have established a strong association between elevated amylin and neurodegenerative diseases and even metabolic disorders like diabetes mellitus type 2. Most relevant studies focus on amylin's neurotoxic potential. Maybox and Fusen in 1999 established that amylin's phenotype plays a large role in its neurotoxicity, as it adopts a similar conformation to amyloid beta, and found that genotype has no part in this, as rat amylin sequence is 95% similar to human amylin and had no notable cerebral toxigenic effect. Lim et al. Findings also concluded that human amylin having a similar toxin-like mechanism to amyloid beta peptides, where it is also reliant on cerebral aggregation. They further suggested the possibility of the toxigenic peptide sharing cerebral ligands, as co-incubation indicated no increased degenerative effect when both peptides were present. Much like Benlier et al. Study, a Zhao et al. Investigation in 2019 sought to evaluate the association between plasma amylin concentrations and Alzheimer's incidence, where they also found a clear association between elevated amylin levels in neurodegenerative patients. It was concluded that both papers serve as supportive evidence for plasma amylin's potential role in various neurodegenerative diseases. Overall, there has been no further progress on amylin's exact role in epilepsy prior to nor since the paper has been published. Thus, this article serves as critical foundation for further research into this field.
While the study showed valuable insight into the differences in serum amylin levels between epileptic and non-epileptic individuals, it does not go without its limitations. One limitation is the study's small sample size, which included samples from only 45 epileptic patients and 60 non-epileptic patients. This sample size may not be large enough to provide an accurate representation of the population. Future studies would benefit from a much larger sample size in order to draw accurate conclusions. The second limitation was the failure to stratify epileptic patients based on subtypes. Future studies could therefore investigate whether a difference in serum amylin levels exists between the subtypes of epilepsy, For example, between patients who experience focal onset seizures and those who experience generalized seizures. This could provide more insight into the link between amylin levels and epilepsy. The study also did not determine the significance of toxic amylin oligomers and amyloid deposits in relation to neural function. Therefore, future studies should include the relationship between amylin aggregation mechanisms and the specific onset of epilepsy.
The formation of amyloid plaques in the brain is commonly associated with the onset of neurodegenerative diseases such as Alzheimer's disease. Disorders such as seizures can be triggered by changes in cerebral neuron conformation and function. Thus, the significance of amylin aggregation in the brain must be determined in neurodisorders such as epilepsy. This study showed that serum amylin had a positive correlation with epilepsy. Thus, more research must be conducted to analyze amylin as a biomarker for epilepsy diagnosis. Further investigation must also be conducted to ascertain the role of amylin aggregation and cytotoxic oligomer formation in the pathophysiology of epilepsy, and the significance of epilepsy subtypes with serum amylin. Overall, this study provided valuable information that could assist in the diagnosis and treatment of epilepsy.